This is a spinach leaf. Most plants have green leaves, and this green color results from the presence of large amounts of chlorophyll in the leaves. Leaves may contain additional pigments in smaller amounts that may not be visible to the naked eye. What pigment besides chlorophyll are present in spinach leaves, and what colors of light are absorbed by these pigments? What role do pigments play in the light-dependent reactions of photosynthesis? This is plant pigments. Chloroplasts are colorful molecules found in the cells of plant leaves. These molecules carry out photosynthesis. Sunlight, which is otherwise known as electromagnetic radiation, provides the energy needed for the photosynthesis reaction. We think of visible sunlight as white light, but visible sunlight is composed of red, orange, yellow, green, blue, and violet-colored wavelengths of light. The structure of a pigment molecule allows it to absorb some wavelengths and reflect or transmit others. We are going to use a colorimeter and a spectrometer to see which wavelengths of light are absorbed by chlorophyll. When chlorophyll absorbs light of a specific energy and wavelength, the electrons in the pigment molecules jump to their excited state. These excited electrons and the electron transport systems within leaves are what make the photosynthesis reaction possible. The ability of chloroplasts to absorb certain wavelengths of light drives the building of a food chain for almost every living thing on this planet. This suspension is a pigment extract made from spinach leaves. I cut up some spinach leaves into small pieces and let them soak in 70% ethanol for about an hour. Then I crushed the leaves with a mortar and pestle. I filtered the crushed leaf suspension through a coffee filter. Like I said before, leaves may contain additional pigments that may not be visible. In part 1, we are going to use paper chromatography to separate out the pigments to see which pigments besides chlorophyll are present in spinach. And then we will use the colorimeter and spectrometer to see how pigment molecules respond to different wavelengths of light. Let's start with chromatography. I made a pencil line about 2 centimeters from the bottom of this chromatography strip. I'm going to saturate the paper along the line with pigment extract. I have to wait for this line to dry, then I'll add another layer. Now my paper is dry enough to add the second pigment line. And now I can carefully set the paper in a chromatography chamber with acetone as the solvent, allowing the solvent to crawl up the paper, up to the pigment line. Be sure to answer the question about the solvent in the procedure. The pigments need time to separate, and I can't disturb the chamber while the separation is taking place. So I'll set this chamber aside for now. While we wait for the pigments to separate, we can see what colors of light are absorbed by chlorophyll with the colorimeter and with the spectrometer. I have already calibrated the colorimeter, and a calibration blank is filled with 70% ethanol, since that was the solvent I used to make the pigment extract. I left some room in my sample just in case I need to dilute it with more ethanol. And I will need to dilute with more ethanol if the sample absorbance of any color is much greater than 2. So now I'm putting the sample in the colorimeter, and I'm going to SparkView, and I'm going to start collecting data. So I scale my data to make sure I see all of the information for all of the colors. And it looks like my sample is okay, since the highest absorbance of any color is right around 2, so that's all right. And the remaining colors are all below 2. For now, you can ignore the table over here on the right and just look at the results for the 6 colors of specific wavelengths in this bar meter graph. You can assume the chlorophyll molecules absorb the colors of light with the highest absorbance values. Pause the video while you record your observations of different values for each color absorbed by the chlorophyll molecules in the sample and answer the questions in the lab procedure. Then resume play when you are ready to move on. The colorimeter showed absorbance values for just 6 specific wavelengths of light, but the spectrometer will let us see a continuous range of wavelengths in the visible spectrum instead of only 6 specific wavelengths. I've already performed a calibration with the 70% ethanol blank in the spectrometry app. Now let's take a look at the absorbance on a continuous wavelength spectrum in the spectrometry app. As you can see, the peak wavelength values detected by the spectrometer do not necessarily line up perfectly with the specific wavelengths measured by the colorimeter. So the colorimeter gets us in the ballpark of peak wavelengths, but the spectrometer gives us more specific peak wavelength information. To see the wavelength and absorbance value for any point on the line, just add a coordinate and drag the box to read the absorbance and wavelength value for any point on the line, whatever wavelength you wish. Pause the video while you sketch the results and resume playback when you are ready. The pigment separation is not yet finished, so we will continue on to part 2. Before we continue on, I want to see if you can connect the dots between the data you see in the blue and violet region for this pigment sample and a quick demonstration with a blue laser pointer that emits a beam of light with a wavelength of exactly 405 nanometers. Would you expect the pigment solution to absorb or transmit blue-colored light through the sample? Let's see what happens to the beam of light as it passes from one end of the cuvette to the other. Is the light absorbed, or is it transmitted straight through the solution? What conclusion can you make from your observations? Recall that when pigment molecules absorb specific wavelengths of light, the electrons are excited and are then picked up by molecules that act as electron acceptors to move the electron along the photosystems and drive photosynthesis. But the spinach extract I will be using for part 2 required me to stick the leaves in a blender with sugar water, which helped release chlorophyll but also destroyed the spinach leaves' photosystems. So the regular electron acceptor molecules are replaced by a blue-colored compound called DPIP to act as the electron acceptor. When DPIP accepts electrons, it is reduced, which changes it from this bluish color to clear. The rate at which the blue color disappears allows you to observe the effect of light on chloroplasts and photosynthesis. All 3 of these cuvettes will receive chloroplast suspension. Cuvette 1 is a blank which has chloroplast suspension and buffer solution. Cuvette 2 will be placed in the light. It will have chloroplast suspension, buffer solution, and DPIP. Cuvette 3 will be identical to Cuvette 2, but I'm going to wrap it in foil so that it will be in the dark even when I place it in light. I'll switch back to SparkView and calibrate the colorimeter with the new blank. And now we are ready to start collecting data. After I record initial absorbance values, I will set both Cuvette 2 and 3 in front of these lights and measure the absorbance of each cuvette over the next 15 minutes at 5-minute intervals. The SparkView file is set up to display red absorbance, but I'm telling you, if it were up to me, I'd choose something different to display. But I'm not going to tell you what I would choose because that's your job. Since I don't know what you've chosen to display, I'm going to show absorbance and transmittance for all colors. I will share snapshots of the data for each cuvette at each time recording in case you can't collect the data yourself. Now I'm going to add chloroplast suspension to my 2 cuvettes. Make observations of both cuvettes 2 and 3. Pause and resume play when you're ready to move on. I'm going to record initial absorbance and transmittance values now. First, cuvette 2. Next, cuvette 3. And and the next reading will occur in 5 minutes. In a classroom, I would use these flashlights to illuminate the cuvettes, but because of the bright studio lights here, I won't need to use any additional lights. Make observations of both cuvettes 2 and 3 after 5 minutes. Pause and resume play when you are ready to move on. Oh, it's got lots of foil here. I'm recording the absorbance and transmittance values at 5 minutes now. First reading is cuvette 2. Next, cuvette 3. And the next reading will occur 5 minutes from now. Make observations of both cuvettes 2 and 3 after 10 minutes. Pause and resume play when you are ready to move on. I am recording the absorbance and transmittance values at 10 minutes now, starting with cuvette 2. And next, cuvette 3. And the next reading will occur 5 minutes from now. Make observations of both cuvettes 2 and 3 after 15 minutes. Pause and resume play when you are ready to move on. I am recording the absorbance and transmittance values at 15 minutes now, starting with cuvette 2. And next, cuvette 3. During the last 5 minutes of the DPIP experiment, I removed the chromatography paper from the chamber, took a close-up picture, and added it to page 3 in SparkView. Now let's move on to analysis, but don't forget to answer the questions that are written in the procedure for Part 2. You can find analysis questions for Part 1 and Part 2 embedded in the procedure for each part. Review both procedures and make sure you have answered all of the questions. For Part 2, it is your call to decide whether you report transmittance or absorbance, and for what color, and whether you decide to report data for more than one color. All these decisions are left up to you. Think about all the choices you have and make the best decision based on your understanding of absorbance, transmittance, and solution color. Now you have enough information to design your own investigation as directed in the lab, and you can move on and answer the synthesis questions. Good luck with your lab.